If there is anything we can all agree on is that salmonella is terrible. It is a menace to society. It is a burden to the health sector, and it is a financial burden to many food companies. Imagine you've made your samples, they're tested positively for salmonella, and you have to toss them all away. But the main issue lies in the fact that we cannot detect salmonella fast enough. There's also the issue where other bacteria tend to mimic the presence of salmonella. And so it becomes a whole situation. This is where phages come in, or bacteriophages. Now, what are bacteriophages? These are viruses that tend to infect bacteria within a specific host range. And we can leverage these because of their properties. And so they're genetically easy to modify, they're safe for human use and animals and plants, and they are abundant, so they're very cheap. Which brings us to this fantastic image here, which is a TM that I took, and I want to highlight that because it's very difficult to get that image. So I have genetically modified a salmonella phage to insert two genes. One is called monomeric streptavidin, which allows the phage to have a magnetic head, which is what you can see. That round thing at the top is actually a bead. And then it has another gene where it produces something called nanolock. With these two modifications, we are able to detect the presence of salmonella. So how does it work? The phage is added to a solution that could potentially have salmonella. If it has salmonella, it will glow blue. And if there's no salmonella, it will not glow blue, because salmonella can only be infected if it is alive. And so this is very good because you can only identify viable cells. Now, the most interesting thing about this presentation is, as I said before, it is very fast. My research shows that you can detect salmonella in less than 7 hours, where the current methods that we have take anywhere between 28, 48, or even 72 hours. And that's just not fast enough for this fast-paced world. And this is why this is very cutting edge, and it is cheap, and it is abundant, and so it is where our research is going. And so I'm very excited to share this because I recently published a paper, and it shows that we can detect as little as less than 10 CFU in water, which is highly specific. Thank you.